Hi friends, today we will talk about India's first vaccine against dengue, which has been approved by the DCGI. This vaccine is called QDENGA and it has been manufactured by Takeda Biopharmaceutical. As we know that this dengue contributes about 390 million cases across the world in 125 countries, and one third is contributed by India in this global burden. The accelerating transmission is because of urbanization, climate change, and the expanding habitats of the Aedes mosquito. As we know that dengue virus has four serotypes: 1, 2, 3, and 4. When the person gets infected by one serotype, it gives him lifelong immunity against that serotype only. Patient does not get immunity against the other serotypes. And second time when the patient gets infection of the dengue with the serotype other than previous one, the accelerated dependent enhancement comes into picture. The pre-existing antibodies, they bind to the new virus, but they fail to neutralize it. So it triggers a hyperimmune response and patient gets critical or severe dengue. So the Director General of Drug Control Authority, that is DCGI, has granted the market authorization to India's first approved dengue vaccine, and this vaccine is called QDENGA, that is TAK003, and it has been manufactured by Takeda Biopharmaceutical of Japan. So this is the first dengue vaccine which has been approved for use in India. This vaccine is a live attenuated tetravalent vaccine, means it gives immunity against all four serotypes of dengue virus. It is administered as a subcutaneous dose of 0.5 ml, and these doses are given 3 months apart. And it is approved for children more than 4 years of age to the adults who are 60 years of age in India. So this live attenuated tetravalent vaccine gives immunity against all the four serotypes of dengue virus. This is basically built on a weakened dengue virus 2 core, and it is engineered to safely express surface protein for other three serotypes. So it gives a dual immune response and successively stimulates both neutralizing antibodies and cellular immunity across all the four variants of the virus. Initially, in the world, the dengue vaccine, that was old vaccine against dengue, was used. When the patient used to report at clinic, his blood was taken for serology and patient had to wait for the lab result. If the patient gets seropositive, then patient was given 3 doses of dengue vaccine. But this QDENGA virus, this vaccine has eliminated all these requirements. When the patient arrives at clinic, he is administered 2 doses of vaccine. First subcutaneous dose is given on the day when he reports, and second dose is given after 3 months. It can be administered regardless of the previous dengue infection, and zero pre-vaccination serological testing is required. So this before approval of this vaccine, 19 global trials were done about on 28,000 patients, and the DEN 302 trial phase 3 data specifically evaluated the safety and immunogenicity in Indian adults and children 4 to 60 years of age, ensuring the localized efficacy. The 7-year follow-up data also confirmed sustained protection across all the epidemic cycles. The key findings at 7 years birth date: Patient has continued durable protection against symptomatic dengue. There was sustained reduction in dengue-related hospitalization rate, and it also demonstrated cross protection across all four dengue virus serotypes. So, this QDENGA virus vaccine, QDENGA, it has dramatically simplified the endemic administration compared to the previous generation of the vaccine, as it is a live attenuated. There is no prior requirement of infections. Pre-vaccination status is not required. It is only administered as 2 doses. It gives a coverage against all the four subtypes of dengue virus, and it is given in the age bracket of 4 to 60 years of age in India. It has a WHO recommendation for use in dengue endemic high transmission areas, and it is also pre-qualified, granted WHO. It has also been given WHO pre-qualification status, which is a prerequisite that allows the bulk procurement by international agencies like UNICEF and PAHO. So it is uniquely suited for mass immunization due to elimination of serological screening. So the strategic deployment of this vaccine targets highly exposed demographic and endemic hotspots. So the youth, children and adolescents growing up in endemic areas, working people, adults up to 60 years of age living in dengue prone regions, the communities which are hotspot experiencing the repeated seasonal outbreak cycles, and occupational health workers, municipal staff, and individuals with increased exposure risk, they are the target for these QDENGA vaccine. The clinical trials which have been done so far on this vaccine, they demonstrate a highly tolerable safety profile with uncommon serious events. So initially, with the use of this vaccine on in trial basis, the serious adverse events were uncommon across all trials, but self-limiting. Some side effects like injection site pain, redness, swelling, mild fever, headache and muscle aches, temporary fatigue, they were noticed in few of the subjects. So vaccination against dengue by QDENGA must complement, but it should not replace the comprehensive vector control protocol. So vaccination does not eliminate the need for environmental hygiene. Patient and municipalities must continue to eliminate the standing water and mosquito breeding sites. They should utilize mosquito bite prevention tactics, and they should seek early medical care and recognize the severe dengue warning signs whenever the patient develops a fever. So this QDENGA vaccine represents a paradigm shift in India's long-term endemic dengue management. So the core takeaways are: it is a first-in-class, India's first approved dengue vaccine authorized for ages 4 to 60 years of age. It provides comprehensive coverage, protects against all four serotypes via live attenuated, and it requires 2 dose schedule, that is on day 0 and after 3 months. No prior infection screening is required. So it removes the largest logistical barrier to mass vaccination, and it is backed by 7 years of efficacy data conducted on 28,000 plus participants, and it has an official WHO pre-qualification. This was all. Thank you very much.